Okay, so we're looking at proteins. The monomer for proteins is going to be this little guy here. What is this? So this is our amino acid, and there's going to be 4 parts that are crucial that you know. Now, we get to see that word alpha again. We saw it with the glucose, alpha glucose. This is completely unrelated. The alpha carbon here is just referring to the middle carbon. And there's going to be 4 things that are located around that alpha carbon. When we looked at functional groups, I told you that it was going to be really important that you made sure to know what your different functional groups are. So 4 things that go around an amino acid. Let's start with the easiest one, is a hydrogen. What are 2 of the functional groups that you should recognize here? Yell it out. Come on. A carboxyl group and a, not methyl, pretty close, an amino group. Okay, so we've got the carboxyl group or the carboxylic acid group on the right. You'll kind of see those words interchangeably. On the left here is the amino group. The hydrogen is obviously the first thing I mentioned. And then there's this R. And R is just a variable group. That variable group is what's going to give every amino acid its unique properties. So everything else is exactly the same. It's a carbon copy. Pun intended? No pun? I don't know. But that R group is what's going to define each amino acid. We'll talk about those in a second. First, I want to look at how different amino acids come together. We've looked so far at ester bonds, we've looked at glycosidic linkages, and now we have a new fancy term. Again, it just means a covalent bond, which is a peptide bond that forms between 2 amino acids. So we're going to have the OH of the carboxylic acid, one of the H's of the amino group. They are going to dehydrate or condense so that this bond is formed. You should be able to draw this. If you see this, you should be able to recognize it. And so when we look at proteins, the kind of central pattern that you're going to see is nitrogen, carbon, carbon, nitrogen, carbon, carbon. And they'll just continue in that same thing, nitrogen, carbon, carbon. It's always going to be the carboxylic acid with the amino groups that bind. Bless you. Okay? It is not going to ever be 2 carboxylic acid groups together. So they always orient in the same direction. Okay. Let me just double check that I did. Okay. I will mention that. That's good. Okay. With carbohydrates, if you're looking at a simple sugar or you're looking at a polysaccharide, they're both considered carbohydrates. Here, we're going to look at it kind of in 3 steps. The amino acids are the individual monomers. The polypeptides are going to be the strands of amino acids, and it isn't until we have completely folded up that polypeptide and given it a 3-dimensional shape that we're going to consider it a protein. So the protein is the functional unit here. All right, so let's look at how proteins fold. Because if you have an unfolded protein, it's not going to do you any good. There are 4 levels of protein structure. What that means is there are 4 different ways in which proteins organize themselves to get structure. We've got what is called the primary, the secondary, the tertiary, and the quaternary. Or in other words, the 1', 2', 3', and 4' levels. If you see either one of them, they're interchangeable. Okay. Primary structure is simply the sequence of amino acids. You'll see proteins often referred to as being like beads on a string. Okay. The particular sequence that those amino acids go are going to give it that primary structure. So if we take a look up here, maybe if I can unwind this, there we go. Right? So I've got a string of beads. Each one of these beads represents a different amino acid. They're all kind of strung together with that backbone of carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen repeating. But it's the sequence here that is going to dictate the primary structure. Okay? Also note, when you're looking at the primary structure, there's going to be 2 defined ends. One where the amino group is free, that's going to be the N' end, and one where the carboxylic acid is free, that's going to be the C' end or the C terminus. So the N terminus is up here, the C terminus is down here, and we're going to number them from the N end. Okay? So again, if I'm looking at this, beads on a string, one end is going to have a free amino group, one end is going to have a free carboxylic acid group. From there, we're going to get localized folding patterns. And this is going to make up the secondary structure. It is because of the interactions of that backbone. Right? So if I go back a couple of slides, remember that I said we're going to have this NCC form over and over and over again. We've also got an oxygen here. We know that's going to be electronegative. We've also got a nitrogen here that's going to be slightly electropositive. So what can happen is the backbone itself can start to interact with each other. If we look at our example here, it's not the beads that are going to be interacting for the primary structure. Instead, it's going to be this like brown pipe cleaner, right? And so there's going to be 2 shapes that are common in the secondary structure. The first is when it kind of loops around and interacts with another backbone, and you make these little spirally guys called alpha helices. I know, you're going to get really tired of the letter alpha. Try not to get too overwhelmed with the fact that they're everywhere. So we can end up making little spirals or making little zigzags, essentially, that fold back on themselves. It's not the beads interacting, it's that backbone. Okay? So you should know that for secondary structure, the 2 things you need to be familiar with are the alpha helix and the beta pleated sheets. The beta pleated sheets are the little zigzags. When we go on to tertiary structure now, this is where the R groups start to interact. Okay. So there are 20 different R groups. Each of the R groups has a little bit different characteristics, but in general, the way that they can interact is through hydrophobic or hydrophilic interaction. So areas that are particularly hydrophobic tend to cluster together. They can actually have hydrogen bonding, again, because of very weak positive and negative forces on them. They can have literal ionic bonds, where you've got a straight up positive charge and a negative charge, or a disulfide bridge. Disulfide bridges are exactly what it sounds like, 2 sulfurs. And these are caused by cysteine, which is an amino acid, which has a sulfhydryl group. That sulfhydryl group forms a covalent bond in the backbone. That's going to really pull this whole thing into all kinds of neat contorted shapes. So, R groups are going to be defined by their sizes, their polarity. Charge, notice polarity is just kind of a temporary, transient, not as strong. Charge is an outright ionic charge, gain or loss of electrons, and whether or not they contain sulfur. So if you're making a necklace, I want you to picture you're putting on some beads. You have a really, really big bead all of a sudden. That's going to cause a little bit of a kink in the necklace. Then you have a bunch of little beads that are nonpolar, and then you use a magnetic bead. And then you put a bunch more beads on, and then another magnet. What's going to happen with the two magnetic beads? They're going to stick, right? That's really what's happening, is the sequence of those R groups is going to then define the overall folding pattern, okay? Then finally we get to the quaternary structure. The quaternary structure, fourth level structure, is just when you have multiple polypeptides that are coming together. They can be part of the same gene or different genes. So for example, we can look at hemoglobin. Science loves alphas and betas. So again, we've got what's known as the alpha subunit, the beta subunit, and when they come together with some heme, which is an iron-containing molecule, that's going to be what produces the fourth level of structure, and this is the functional protein. Some proteins only have a tertiary level of structure. They don't need extra stuff added into them to make them work. Some do. So this one's kind of optional. And a prosthetic group just means something non-organic. Or non-proteinous. Questions so far? Pretty easy? Manageably easy? Okay. All right, two classes of proteins that we're going to look at. Globular. Globular is in globs. Fibrous is in fibers. I know, tricky, right? By the way, what do you see right here? Arrows, but that kind of like are sheeted up on top of each other. So those are representative of the beta-pleated sheets. And these little spirally guys right here are representing the alpha helices. So usually if you're going to have R groups on opposite ends of the protein that attract towards each other, that's going to make something globular. It's all like whoosh, whoosh, right? Whereas fibrous tend to not glob up so much, but they will bind with other proteins and make long strands. Okay. Yes, you do need to know all 8 functions of proteins, and you should know at least one example of each. I know it might be hard to remember all of those various things, so maybe finding a mnemonic might help you. I'll give it time to sink in. By the way, here's my... Okay, so structural features. Those are things like collagen, right? As women especially get older, they lack collagen and buy really expensive things that add collagen. No, they don't. I mean, they have collagen, but then you digest it, and it doesn't really do anything productive for you. Hormones. Some hormones are lipids. Others can be proteins. So for example, insulin, that is going to be a protein. Your immune system has antibodies that help destroy pathogens, so it's important that you have proteins for that. Transport, such as hemoglobin, is going to transport oxygen throughout your body. Cell membrane proteins are transporting materials through the membrane. Your movement, your muscles, are because of the fact that you have protein. So when bodybuilders are taking like tons of amino acid supplements, that is so that they can get like Arnold style. Enzymes we've already talked about. Notice that they end in ACE. That means it's an enzyme. And most of the time, we'll be looking at proteins. Receptors, that's going to kind of go along with like cell signaling. So we'll see that in Unit 5. And then storage, such as albumin, that can then be converted into other types of proteins later on. And this is my favorite. Proteins have to be in the right environment. And as they're being produced, we'll look at that process later, but as proteins are being made, it's like they're getting made in this long strand, and they have to fold in just the right orientation at just the right time. So we have what are called chaperonins. They're proteins that help with protein folding. And they're very barrel-like. So they basically exclude the outside environment and just let the protein interact with itself in a way that it will fold properly. Everything about protein comes down to its shape. If it is the wrong shape, it will not function. That's bad. So let's take a minute and take a look at what can happen if your proteins get screwed up. This is my favorite stuff right here, okay? First of all, if you have a properly folded protein that unfolds, that's called denaturation. That can be due to high temperature, high pH, that's not ideal. Remember all of those ionic and hydrogen bonds? Well, if you don't have very many hydrogen ions in your solution, if it's very alkaline, that can disrupt those bonds and cause it to deform. Also, a low pH can do something similar. Too many hydrogen ions can cause the interactions of the tertiary structure to start breaking down. Once you have denatured a protein, the only way to renature it, the only way to fix it, is to have that chaperonin available again. And even that might not do it. So most of the time, once it's denatured, renaturation does not happen. Okay? So, if you even substitute one thing for another, one amino acid for the wrong one, that can cause problems. These are genetic problems. If you have the wrong genetic information present, you will not make the functional protein. Okay, this is my favorite. Let's talk about prions. Does anyone know what a prion is? Has anyone heard of prion disease? The most famous one is technically known as bovine spongiform encephalopathy, also known as mad cow disease. What happens is the proteins around the neurons start to shape incorrectly, and their misfolded shape affects other proteins nearby and causes them to fold incorrectly, which then basically spreads. So it's a disease in that it can spread, but it's only a protein. It's not an entire living organism that's making you sick. It's just a misfolded protein that causes other proteins to also be misfolded. Okay? If you have a whole bunch of proteins that are misfolded, not because of a prion, right? Not because of one thing causing it, just it's not getting folded properly in the first place, those can accumulate and cause things like Alzheimer's. So too many misfolded proteins in your brain tissue especially can lead to neurodegeneration. And then denaturation. Let's say you've got a fever. It's like 104 degrees, 105 degrees Fahrenheit, not Celsius, because that would really suck if it was Celsius. 104, 105. What starts to happen is your metabolism ramps up. You start having more and more chemical reactions happen faster and faster. Why? Because as you heat something up, it reacts faster. And you can actually get to the point where your fever is so high, 108, 109, 110 degrees Fahrenheit, where now your proteins start to denature. You literally cook yourself from the inside outward. And, you know, that makes you die, in case you were curious. Yes. Okay? And I don't want to do this. So, yes, I do. I should, shouldn't I? I should mention this. Okay. I just want to look at X-ray crystallography and bioinformatics because they represent kind of the old school and the new school ways of understanding protein structure. It used to be that you would take proteins and try to crystallize them, right? Once they were crystallized, you would shoot a laser beam. I like laser beams. At it, and the direction of particles that would bounce off from the laser gave you an indication of what that structure was. And it usually looked something like this, and it took X-ray crystallographers weeks to decipher protein structure. Now what we use is something called bioinformatics. We take the DNA that makes the protein, sequence the DNA, then know the structure of the protein that results. Obviously, one is far more accurate. Obviously, one is far easier to do, although there is a little bit of complexity added by the fact that sometimes we don't know what parts of genes actually get expressed, but we can talk about that later. And with that, my quick lecture is done.